How about that? Can you guys hear me okay? And just do this whenever I start to talk fast and low, which I will do. Singularity is really all about time compression, getting a lot done in a little bit of time. So we're going to run through about 500 slides here in the next 15 minutes. So this is who I am, and I usually just flash this up to tell people I went to school, I worked a couple of places, trust me, but I want to point out because I want to talk to you guys in particular about some work that I did when I was at a little company called Illumina. Has anybody ever heard of Illumina? Excellent, excellent. So Illumina is a life sciences company in the US. They've been known for competing with Affymetrix. They make biochips. They make chips where you go ahead and you look at aspects of your genome, and it's very fun, it's interesting, it's sort of like a cross between a computer chip and DNA, but it only lets you look at some of the DNA, and I wanted to talk to you guys about that a little bit. So this is the intersection of what I work on. I do a lot of work in personalized health. I've got a startup company called Genomera. I work at a couple of nonprofits. I get to work with these guys every day. And let me talk to you a little bit about DNA sequencing. So, in a lot of ways, whenever you look at biology, it's about answering the big questions. How did life begin? How do we get from here to more of an idea that's scientifically accurate? How do organisms grow and develop? How do we get this from this? How does consciousness hook into matter? The big philosophical question. How can a wet computer control a muscle and bone effector and produce something like this? And what's the basis of life's blueprint, which is actually, unlike all these other questions, a question that we've now answered. Because we know about DNA. About 50 years ago, Crick and Watson and Franklin, don't forget Rosalind Franklin, solved the structure of DNA, and it's had these great implications, and we've developed a lot of tools to let us look at that structure. We've made these microarrays, which are these computer chips, and here's a chip about the size of a microscope slide that lets you look at roughly 1.2 million places in your DNA. It means that there's an array of 1.2 million spots on here. Each one's got a different little piece of DNA, and you can use this to see how your DNA is different from my DNA, different from somebody's DNA who is going to develop cancer when they're 10 years old. So this is a really interesting thing. And it's not just interesting from a research perspective, but it's interesting now from a consumer health perspective, because there's a company called 23andMe that will not just sell this to doctors or research institutions, but will sell it to a consumer who's got a Visa card. For $99, you can get all this information interpreted in really friendly language, what we call genomics for grandmas. So, and my mom, who is 73 years old, reads her profile and she'll call me up and she'll say, honey, there's a new research article, and I wasn't sure if this applied to me or not, but it looked like it was a 3-star article. They've got a little rating system, and this is something I'm interested in. So it's something that is available today. But sort of the next step, instead of looking at 1.2 million...
Can you please...
Oh, thank you very much. Instead of looking at 1.2 million pieces of DNA, am I still hard to... Let me go ahead and try this. Is this okay? Thank you. And let me know. I don't mean to do that. Instead of looking at 1.2 million pieces of DNA, what if you could look at all 3 billion bases that make up your human genome? And so we've got a machine that does that. And again, there's a little thing about the size of a microscope slide. It's got these channels that are grooved into it. And there are holes at each end, and you go ahead and you stream in DNA, and it makes a lawn of these little short bits of DNA, single strands of DNA. And what we do is we build up the other side of that strand of DNA with colorful bases, different color for each base, a different color for C, G, A, and T, the alphabet of DNA. And we look at the spots that makes under a really powerful charge-coupled device. It's the same thing that they use in the Hubble Space Telescope, but we're not looking out at the stars, we're looking inward. And we do that. And we've been doing that, and we've been doing that exponentially. Here's a graph, and like a lot of the things that we talk about at Singularity, this is an exponential increasing graph. You see that it's got a logarithmic scale, so one leap up on there is 10x improvement. And this is a graph of all the DNA data that had been stored, had been sequenced and stored in the world. And it goes up and up and up until 2008, whenever we invented this next generation sequencing machine. Here is a synopsis of some of the genome sequencing projects, starting with the Human Genome Sequencing Project, which ended in 2001 and cost, depending on whose figures you want to use, $300 million or maybe $1.5 billion. Took 13 years to finish. And then this year at Singularity University, we're going to be using a couple of different machines to sequence some of our students who are volunteering for $2,000 a piece. It won't take 13 years. We're going to do it over the summer. So here you see, over a little more than a decade, a better than 500x improvement in the throughput on this. And people always ask, how long is it until we get the $1,000 genome? Or I like to think, how long until we get the $1 genome? And what do you do whenever you have that? And I want to tell you real quick about one scientist, his idea. He said, we will sequence everything. If it tastes good, we'll sequence it. If it's cute, we're going to sequence it. And we hope he was talking about the panda looking cute and not tasting good. And this was in response to somebody had challenged him. He had a really ambitious project he wanted to do for China called the Tree of Life Project. It had 50 different animals in it, 100 different plants. It had this tremendous number of things. And at the time, people said, you know, that's going to take 40 years to complete. It's going to take billions and billions of dollars. Well, cut to the future, Illumina makes this machine and is getting ready to sell it to people that's able to do very quick genomics. And Jun Wan, who had been at this conference, comes and buys 128 of them for $500,000 a piece and puts them in the Beijing Genomics Institute. That graph I showed you earlier that showed all the genetic information that had ever been sequenced in the history of the world up until 2008, he can do all of that every 15 minutes now. And this is where they do it. This is an installation where each of these $500,000 machines, they're just putting them in like slot machines in a casino, plugging them into the floor. And like slot machines in a casino, we're not sure why they have the eye in the sky up there, but they're keeping a close watch on this. So one of the questions people ask is really, how long is it till the $1,000 genome? And you can graph out what scientists pay to do this. You can graph out what retail people pay to do this. And again, notice this is one of those logarithmic graphs. And if you extend this and look, for very good reasons, we think this trend is continuing, getting cheaper every year at about 5x. You get the ability to do a human genome for about the cost of a chest x-ray in the next 2 years. 2 years after that, you do it for cheaper than a delivery pizza. And by the end of this decade, the cost of a human genome is less than the cost of flushing a toilet. And one of the things I threaten people with is we're probably going to do a genome every time we flush a toilet because there's really interesting biological data in there. But no one likes to think about that. So a penny a genome in less than 10 years. Somebody who's going to school now to study this, who is going to maybe come out and be a PhD or a medical doctor, by the time they get out, a penny to do the whole human genome. And so what? People want to know, well, you know, why does that matter to me? Let me just talk to you a little bit about that instead of running through a bunch of slides. So why does that matter to you? We've got really good data about why my genome is a little different from yours and what that means. And as an example, Let me just skip to something I want to show you. We've studied drugs and the proteins they target really well. We understand when people have these slight differences in genetic data, what it means. And here's an example. There's a drug called Plavix that's a blood thinner. People take it to control, different chronic conditions. But if I had a heart attack, dropped flat on the floor, they would send an ambulance crew, and on the way to the hospital, they would give me Plavix. That would greatly increase my chances of surviving a heart attack, except for me, it wouldn't, because I have these 5 differences in my genome than most of you here. I would need 3 times as much Plavix. And an ambulance crew won't know that. And until we have good electronic medical records, this is how I tell them. I wear around my neck a little medical alert piece that has all of the information that would tell them, this guy, if you give him Plavix, either give him 3 times as much or give him this different drug, Effiant. So here is a place where genetic information could save my life. And I'm a little ahead of the curve on this, but I did this back about 5 years ago. 2 years after that, 23andMe started featuring this kind of data. And now you see this kind of data. Doctors are doing the same thing. They're saying, hey, here's a patient I have who's at high risk. We ought to make sure this is in the medical record. We ought to go ahead and make sure their family members know if they need to get this drug, this is what they need to do. So we're in an era of very cheap sequencing. And as entrepreneurs, as people who are trying to improve the world, if we can't do something with this, something is wrong with us. One of the things I wanted to mention too is, this is all about reading DNA. The really stunning things start to happen whenever we can write DNA. And we can actually do that now. And I have a whole slide set I would love to show you on that didn't load up. So let me wave my hands a little and talk to you about that. Once we can write DNA, and I'm not talking about Gattaca, I'm not talking about genetic engineering. Once we can go ahead and make changes in DNA, the first changes we will make are not so much in you and me for medical reasons, because that remains hard for now, but the changes we'll make in microorganisms. We'll take E. coli and splice in genes from other organisms. And we do that now whenever people take insulin. If you know a diabetic, that insulin was not extracted as insulin from some animal like we used to do it. It comes from the cells that have been genetically engineered to produce it from a bacteria. And we're going to see that more and more, and not just for medicines, but for manufacturing. The future, in a lot of ways, starts to look like a big brewery vat where we make our materials that we use, our plastics, where we go ahead and produce the drugs that are important to us, where we go ahead and produce the things that we use to fuel the energy economy, and whether those are biofuels or other ways to harness sun power. So synthetic biology, this idea where you can go ahead and take genes and plug them into a cell and build basically a circuit and a machine that produces what you want, is going to be a big driver in the world economy. So I'm going to leave you with me not suffering a heart attack and the idea of synthetic biology, and I would love to take some questions and talk to you.
Thank you, Raymond.